Okay, so we've been covering this topic of homogeneous constitution, and we have some more words to say about it here. So homogeneous constitution does not mean that every cell in a culture are equivalent genetically or phenotypically. It implies the following. A culture of mixed population of cell types will eventually reach a steady state level of each population within the culture, and these steady state levels will not change when subculturing if culture conditions are kept the same and the passage number is low. So subculturing is simply the whole thing of passaging cells and going from one plate of cells or one dish of cells and going to 4 or 5, right? So you're starting out with that initial culture and the cells grow and you split them, and that process is called also subculturing. Passaging, subculturing, splitting, they're all the same thing. Okay. And we've talked about this. Each time a culture is subcultured, its passage number is increased by 1. The passage number of a culture is an indication of how old it is. The larger the passage number, the more likely that culture is no longer equivalent to a culture of the same cells at a lower passage number. This is something that we covered in the previous video in more detail. A culture of a single cell type will have different populations of that cell type that are slightly or dramatically different from each other. These differences originate from differences in the genotype, which extends into the phenotype of these populations. These populations will remain at a constant levels at a low passage number. Thus, each subculture will be equivalent. Again, these are things that we talked about in the previous video. I just written down here for you guys also. So tissue culture is also economical. Less reagents and equipment are needed to maintain cell cultures than animals. So keeping animals requires equipment, rooms, or spaces that are specifically designed to maintain and keep those animals. And it tends to be much more expensive than simply doing work in cell cultures. So much less experimental drugs or expensive reagents are needed in tissue culture system as compared to an animal system. So what this means right here is that if I need a certain concentration of a drug or a reagent within an animal or an organism, I will only need a fraction of that in cell culture because I will get to that same concentration very quickly because there's less volume. Many legal and ethical questions are avoided when using a tissue culture system instead of an animal system. And this is important because anytime you do animal work, you have to justify it, and you have to justify that you're using humane conditions. So, if something can be done in a tissue culture system, there's no need to do it in an animal system. You move to an animal system where the tissue culture system does not exist, or you've done the work in the tissue culture system and now there are specific questions you want to have answered that requires a full organism. You do not take the questions that can be answered in tissue culture and just simply move them to an animal system because there's all these other considerations. It's more expensive. It's more time consuming too. So if the question can be answered in a tissue culture system, you go ahead and answer it in a tissue culture system. And then you need certain questions answered in a full organism. You move on to a small animal model or a large animal model. Small animal model being, for example, mice or rats. And a large animal model will be something like non-human primates like monkeys and chimpanzees and etc. So limitations and disadvantages of tissue culture. A level of comfort and expertise needed to be obtained by the experimenter. And this was actually something that you guys would have been picking up if we had the lab on campus, but unfortunately that is not the case this semester. This is not a big hurdle. It is something that you learn very quickly. It's just one of those things that a little bit of experience doing it, you pick it up. Can be expensive if the end goal is mass production of tissue or cell products. So if you are working in large amounts of cells, then this can become expensive. Typically in a laboratory environment, you're not doing that. The other problem is artificial selection, and this is different than natural selection because, well, it's artificial. Cells taken directly from an animal will contain many different cell types. So if I go in and I pick a little bit of tissue out of an animal, within that tissue, there will be different cell types. It could be that the majority of cells that I pick up are the cell type that I want. For example, if I take tissue from the liver, from where I took it in the liver, it could be that a majority of those cells in that tissue are hepatocytes, which are the liver cells, right? Within that tissue will also exist resident immune cells, fibroblasts, and etc. These are other cell types that are in the tissue that support the organ and the tissue itself. The cells that are most fit in the culturing environment will outcompete other for resources and therefore be selected for. If I create an artificial environment within the petri dish, within the plate of cells that I have, and I have all these different cell types. So here's my plate of cells, and I'm just going to put different cell types, denoting them being different by the color. Okay, so here's a couple of cell types. Here's one, it's in red, and let's say I have another one in green, and the green is, a minority here, and I have blue. Okay, and maybe a couple more blue. Here we go. Okay, so if the solution that I bathe these cells in tends to favor, for example, the green cells, as I passage these cells, meaning that I split them and I subculture them, eventually what I'll get to is all green cells. Why? Because the green cells are in an environment that works really well for them. And they will outcompete the other cells for resources, so the other cells will simply die and not be able to replicate well and eventually die off. But the green cells are doing great, and they are replicating and they're thriving in this environment. But what if I don't want the green cells? I actually want the red cells. Those are the cells that I want. So I have to create an environment that favors the red cells over the green and the blue cells, so that when I passage them, then if my solution and the conditions that I'm keeping the cells in favor the red cells, what will happen is, after those many passages, I will only have the red cells in my culture. We'll say this is the first one, condition 1, and conditions 2. That could be anything from temperature that you keep the cells in, but typically it is not temperature, but what the solution that you're bathing the cells in contains. Thus, it is important to optimize the culturing conditions to either maintain the proportion of different cell types or favor a certain cell type, or the technology does exist to physically separate the cell types of interest from the mixture. So the process of dedifferentiation. So before talking about dedifferentiation, we're going to talk about differentiation. Okay? So differentiation, all cells in an animal originate from a fertilized egg. The fertilized egg zygote and the initial resulting cells from the first few rounds of cell replication are pluripotent. These cells are typically called embryonic stem cells, and they can become any cell type. The process of differentiation is typically a one-way street, as in once a pluripotent cell commits to becoming a certain cell type, it cannot reverse the process. During the process of differentiation, some genes are permanently silenced while the expression levels of others are adjusted. Due to the silencing of genes and adjustment of the expression levels of other genes, a unique and specific phenotype is achieved. This results in different cell types within a multicellular organism being very different than others while being genotypically equivalent, meaning that they contain the same chromosomes, all the genes are still there, but the phenotype is very different. So let's take a little bit of a closer look at this. All right, so we start out with an embryonic stem cell, right? Or embryonic stem cells. So the embryonic stem cells, during the process of development, will start to split off in different differentiation paths. And we'll just call these A, B, C. Now, it doesn't stop there, right? During the initial rounds of cellular replication, as the organism is developing from a few cells to eventually become the multicellular organisms it is meant to become, The differentiation process, for example, going down path A, this could still split off. A could split off to be multiple cell types. To not complicate this too much, we'll just say that these paths all split off into two other paths. And then what you get is A1, A2, B1, B2, C1, C2, right? And these can split off even more, and you get A11, A21, A22, B11, B21, B22, C11, C12, C21, C22. Now let's say that these right here, the end, all these cells right here, this is the fully differentiated cell types. And these will make up the organism. One of them could be hepatocytes, liver cells, the other could be some type of immune cell, the other could be neurons, a specific type of neuron, and etc. This is a very limited example. There are many different cell types within an organism, and this is just showing a few, right? So this process of differentiation, meaning that you have these different paths and you get these different cell types that eventually will form the organism, this process is called differentiation, and it's not reversible. Like, you cannot go from A11 back to A1 and then back to A and back to the embryonic stem cell. This will not happen naturally. Yes, it can be done in a laboratory, but that's artificial. At least the technology is starting to exist where you can roll back the differentiation status of adult cells or fully or differentiated cells. But in the organism, this is not going to occur. So what is happening? How do you get a specific cell type? So the phenotype of all these terminally differentiated cells is different than the embryonic stem cells, and they're different from each other. And what you have happening is when I'm comparing two different cell types, let's say I have an organism that only has 5 genes. Let's just imagine that exists. And my y-axis is going to be level of gene expression. Basically, how much RNA is made, messenger RNA is made from these genes. Okay, so that's what we mean by level of expression. I know some of the y-axis on the left one is off screen, but it's the same as the one on the right. Okay, so I apologize for that. So let's say that we have gene A, gene B, gene C, gene D, and gene E. And in this one, we also have the same thing. These are two different cells. Two different cells that have different phenotypes. Okay, so imagine one could be a neuron and another one could be a kidney epithelial cell. Okay, so cell type 1 and cell type 2. Now, when we're looking at the expression levels of these different genes, we can see that they'll be different between the two cells. So, for example, it could be that A is expressed at the same level in both cells. Why? Because maybe this is some sort of housekeeping gene. What do we mean by housekeeping gene? Imagine the enzymes that are involved in cellular respiration. All cells need to do cellular respiration. So those enzymes will be expressed in all cells. So we term those as housekeeping genes. So certain genes will be expressed in all cells because they're important to maintain life in all cells. It could be that B is expressed in cell type 1, but it's not expressed in cell type 2. Whereas C is not expressed in cell type 1, but it's expressed pretty well in cell type 2. Now, this is the case of, in cell type 1, B has not been silenced, where in cell type 2, that gene has been silenced. Doesn't mean gene B is not in cell type 2. The gene is still there, but it has been silenced, so it's no longer being expressed in cell type 2, where it's being expressed in cell type 1. Now, it could be that D is being expressed very well in cell type 1, where it's being expressed not so good in cell type 2. Again, this is differential gene expression. So D is kind of partially silenced, if you want to think about it in that way, in cell type 2, where it's being fully expressed in cell type 1. Now, E could be partially expressed in cell type 2 while not being expressed in cell type 1. Meaning that in cell type 1, E is silenced, where in cell type 2, it is being expressed, but not expressed that much. And this differential gene expression leads to unique phenotypes, right? The phenotypes we see, the phenotypic differences we see between different cell types, okay? And that's the process of differentiation. Now, if I were to take a cell, let's have another example. Okay, so imagine that I have a cell and my Y and X axes are the same. This is gene expression levels on the Y axis. And I have genes A, B, C, D, and E. And I look at the differential expression and I see that, okay, I have a little bit of A, I have a lot of B being made, very little C, no D, and some E, right? Now, when I move this, now this is in vivo. So let's just make sure we know that. This is in vivo, within the organism. When I move these cells to an in vitro environment, an artificial environment, what will happen is that the expression levels don't necessarily change. They might change a little. Why? Because the expression level of certain genes is dependent on the stimulus that that cell is receiving from its environment. So let me draw this out to make it a little bit more clear. So here's my cell. In the in vivo environment, it's getting different stimuli. So it's getting different stimuli, and I'll have them in different color. Stimuli 1, stimuli 2, stimuli 3, and 4. This could be hormone molecules. This could be just signals that cell is receiving from cells right next to it, and etc. And this is in vivo. When we move the cell to an in vitro environment, well, this is an artificial environment. And typically, this is going to be very different than the in vivo environment. There's really no way to replicate the in vivo environment in vitro. Not completely, okay? There are culturing methods that attempt and they get close to the in vivo environment, but it's never going to be the same, exact same as the in vivo environment. So in the in vitro environment, yes, the cell might be receiving the stimuli 2, but it's not receiving it maybe at the same amount. So it's a little bit less compared to the in vivo environment. Maybe it's receiving signal 1 at the same intensity as it was before. And it's receiving 3, but maybe it's receiving 3 at a much higher level than it would in vivo, and it's not even receiving 4. So what you're going to have is, because the signals coming into the cell are different in the in vitro environment, the phenotype of the cell will change. And sometimes that phenotypic difference comes from gene expression. Imagine that the signal A coming into the cell here, right here, right? Imagine that leads to expression of E. This leads to E being expressed. Now, this is fine because it's getting the same level of signal in both cases. But imagine that 2 leads to B being expressed. And this is very typical. Sometimes transcription and translation does not occur until a cell receives some stimulus from its external environment or internal environment. So if this is the case where B is under the control of the stimulus 2 being received by the cell, then if we go above, we can see that, okay, well, let's say that A does not change when we're in vitro. But B is down. C is the same. Of course, D is silenced. And E is the same. So now you have a slightly different expression level of different genes. What you will get is a slightly different phenotype. So this phenotype is going to be slightly different. The in vitro phenotype will be slightly different than the in vivo phenotype. Now, these two are not going to be exactly the same. Does that mean that if I took a liver cell out of an organism and cultured it, and I have a culture of hepatocytes, that they're no longer hepatocytes? No, because you still have this. You still have, for example, in this case, D silence, right? And of course, there's other genes that are permanently silenced. There's other ones that are partially silenced. So that still is there. That fingerprint of being a hepatocyte is still there. Or that fingerprint of being a neuron is still there if you're culturing neuronal cells. That fingerprint is still there. But the phenotype of the cell, that hepatocyte, for example, if you took it from the in vivo environment to the in vitro environment, those hepatocytes, their differentiation status does not change, but their phenotype will slightly change. And the more dramatic the difference is between the in vivo environment and the in vitro environment, that phenotypic change could be a little bit more. But you still have the characteristics of that original cell type. An example of this would be, and this is actually a very good example to highlight what I mean by this. Again, a virus example because I'm a virologist. There are viruses that we know exactly what cells they infect within the organism. So we have a virus, we know what cell it will infect, cell type. So let's say cell type 1, in vivo, and of course the cell gets infected and makes more virus. New virus. But the same virus, if I take these cells and I move them to an in vitro environment, same cells, I'm not changing the cell type. I get nothing. No new virus is made. Okay? Why is that? Well, when I transferred those cells, that cell type 1, into an in vitro environment, its phenotype slightly changed, and that change was enough, so now those cells cannot be infected by this virus. And this happens all the time. It actually hinders the study of viruses because you don't have a tissue culture system for that virus where you can infect cells and study how that virus infects cells and etc. That was actually the case for HCV for a long time until the problem was solved later by finding a unique version of the virus that infected cell-cultured liver cells, and that really propelled the field forward to a point where we have pretty good drugs against HCV. So this process of the phenotype slightly changing is called dedifferentiation. So dedifferentiation does not mean I'm rolling back the differentiation status. All it means is that the differentiation status still exists, but the phenotype of the cell has slightly changed when I bring it into an in vitro environment. So the process of dedifferentiation is not reversing the differentiation decision. Cells are constantly responding to environmental cues, which include signaling molecules within the extracellular environment, connections to basement membrane proteins, physical parameters such as temperature and pressure, and chemical parameters such as concentration of oxygen and pH. The response by cells will include gene expression within the confines of its differentiated status. And all this means is that you're not going to express genes. The cell is not going to express genes that have been silenced. Other responses which do not require the expression of genes. If you're thinking about responses which do not require the expression of genes, the motor neuron excites the muscle cell to contract. There's no new gene expression. There are responses by the cell to cues from other cells or their external environment that do not require gene expression. Thus, a major contributor to a cell's phenotype are those responses which are caused by the environment a cell is in. Thus, removing a cell from its in vivo environment will lead to its phenotype changing, sometimes drastically. This process is called dedifferentiation and is only important if it will affect the experiment being conducted. Example 1. If a researcher is studying the effectiveness of a potential cancer drug on cellular mechanisms within cancer cells, he/she has to make sure that the cellular mechanism is not overtly affected by the culturing conditions. Example 2. If a researcher is studying the replication cycle of a virus, then he/she has to make sure that the susceptible cells can be infected by the virus while in culture. So, dedifferentiation, it really only matters if it's overtly affecting the system being studied in the cell. If it's not affecting the system being studied, then there's no reason to really worry about it. You can still do your study. You know that the cells that you have in vitro are not exactly like the cells that you would have in vivo. If your study will eventually grow to see whatever you're studying that it's happening in vivo, you start out with cell culture and you move up to the in vivo small animal model studies eventually. So, and during the in vitro, all you care about is that, okay, that system that I'm studying within the cells is intact and it's responsive. And that's all. Okay, guys, we'll stop here and continue later. Bye-bye.